Okay, I think I'm through. You please go ahead. I take this opportunity to wish you all a very enjoyable 4 days of discussions, meetings, and learning for everybody for the betterment of individuals, the institution, the society at large, and India. Jai Hind.
Thank you, sir.
Sorry,
Sorry,
I got
there was some technical glitches in between, so sorry for that. And thank you very much, sir, for your golden and kind words. You always enrich us with knowledge, and your talk is always very informative. I think someone is mute. Please mute yourself. Now, I would like to invite on this virtual dais our chief guest of today's function, Dr. Mukul Jain, sir. He is president at Zydus Research Center, Ahmedabad, and is heading the non-clinical research and development group. He is also a lab director of CAP and ISO certified CleanPath laboratories and chairman of many institutional animal ethics committee. He was the key person involved in the discovery and development of saroglitazar molecule. He has also contributed in the discovery and development of around 15 NCEs that has crossed the IND stage of development. Besides NCEs, his group completed safety assessment of 14 biologicals and 13 vaccines of Zydus Cadila. Dr. Jain has obtained his BPharm, MPharm, and PhD degree from Nagpur University, and he has also done executive management course from IIM Ahmedabad and did his postdoc at University of Florida, USA. With over 30 years of professional experience by working in companies like Vocart, Ranbaxy, University of Florida, and NIPER, presently he is associated with Zydus Group since 2000. He has to his credit more than 156 full-length research papers, 4 book chapters, and over 60 patents. He is member of governing board of NIPER Ahmedabad, editorial advisory board member of ACS Chemistry of Letters, and member of various international scientific societies. With such an extraordinary resume, I invite you, sir, to say a few words on this occasion.
Thank you very much, Dr. Kalro. Mr. B.M. Shah, Dr. Chavaria, Dr. Yamini. Let me begin by thanking you all for organizing this very interesting international conference on biologics. I also take the opportunity to thank other members of organizing committee who are not present here, but they were instrumental in putting the things together. Because of the virtual nature of this meeting, we cannot see everyone, but I'm sure this kind of event needs a lot of efforts. I take the opportunity to wish a very good evening to all the distinguished speakers who are participating in this event, all the conveners, researchers, students, and the guests who are connected online to attend this meeting. And I'm reminded second time by Professor Kalro's mention about Murphy's Law that whatever can go wrong does go wrong. So I think we have to prepare for that. When things go wrong, not only in the computers, but also in our society. And I think last year has been, has witnessed unprecedented time for unprecedented measures that happened because of the COVID. And but we are very innovative and resistant to such kind of challenges that comes in our way. And whatever hurdle comes in our way, we try to find solutions. And in fact, it has become really comfortable these days to attend virtual meetings. It was unthinkable earlier that we would organize virtual meetings of this kind, where you have hundreds of people participating, so many distinguished speakers. Many of them are actually based in US and other countries, are joining the same event together by sitting in multiple continents and multiple locations. And I think this is something that we have learned from the challenges that comes in a way. Now, I think for this meeting, it's really great pleasure to address this event on the theme of biologics in healthcare. And at this time, biologics, I believe, can't be more relevant than in today's scenario, because the world is going through this COVID pandemic. And it's been a year now, and the biologics in the form of vaccines are already there to the rescue. 2 vaccines already available for the use, and there are many vaccines in the pipeline and soon going to come for the rescue, and would be available to protect everyone from this COVID pandemic. We know pandemics have happened in past as well. I mean, Spanish flu, which is said to have infected about a third of world's population at that time, and so many people died. But I think today the scenario is changed. What is different today is that we are better equipped with cutting-edge technology, including biotechnology, and which can guarantee that pandemic can be contained as soon as possible. We know that fight against COVID-19 is multi-pronged today. We've been working on multiple frontiers, right from antiviral drugs. People have been using convalescent plasma, monoclonal antibodies, and now the latest to join the armamentarium is the vaccines. And this is the result of a continuous evolution of biotechnology and the science in general, that we are able to have this kind of therapeutic agents. And vaccine has really seen a lot of changes in the last so many years, right from being developed as a live attenuated vaccines to inactivated vaccine, then antigen-based vaccines, and viral-based vaccines. Now, the latest to join are the nucleic acid-based vaccines, which are truly the outcome of the biotechnological advancement that we are seeing today. Moreover, now the vaccines are being looked at from a therapeutic perspective. I mean, we are today talking about COVID because of the nature of this disease, but the vaccines, as the science is evolving, are also being developed for treatment of diseases which have nothing to do with infections, and those are the kind of therapeutic vaccines which are also likely to come in the near future. And it won't be very long before we will have such therapeutic vaccines. Things have changed in the biotechnology front dramatically. I think in my lifetime, I'm seeing those things happening, which is really remarkable. Humulin was an important milestone in the therapeutic biologics, and that was the first ever recombinant biotech-based product that changed the therapeutic landscape. And then number of other kind of biologics came, and the evolution continued to bring out monoclonal antibodies, which were first the murine antibodies, and then we had the chimeric antibodies, then we had the humanized antibodies, and then we are talking about modified humanized antibody with different size and shape, which give them distinct properties and advantages, and they are suitable for specific indications. We also saw evolution of monoclonal antibodies to next level of designer kind of drugs that are monoclonal antibodies loaded with armamentarium, that is like ADCs, those are antibody drug conjugate, which is a combination of biotechnology with the small molecule chemistry to give a new kind of therapeutic agents. And examples of them are already there in the therapeutic use. Same way, I think for virus therapies, we are also seeing emergence of oncolytic viruses. They are modified viruses, and which can be useful for treatment of cancers. So a lot of things are happening, and the evolution is continuing. And I think we are in an exciting time witnessing right before our eyes a disruption like any other in the advancement of the biological sciences and its impact on the healthcare industry. It's taken a little more than half a century from the learning about the DNA structure, the double helix, to getting the PCR setup now in even the small laboratories. The even diagnostic labs have PCR. It was kind of unthinkable, that we will be using this, one time so-called sophisticated technologies as a routine diagnostic test. When I was doing my research, the sequencing was done only in the national labs, and it was considered as very expensive and exotic science, and it used to take a year to sequence the whole genome of even smaller insects. Now you can do the sequencing of your own self in a matter of days and costing so low a price that one can afford himself to get the complete gene sequenced. So these are becoming very much convenient and optimized. Now, recombinant DNA technology is no longer sounding like a high tech that we used to consider nowadays, but with the CRISPR and Cas technologies, now they are becoming more and more accessible and widespread. We are able to manipulate the genes in a manner that was unthinkable, and we can do whatever we want. We can cut any gene, we can add stage, you can, all these kind of things are so easy to do nowadays. And with these advances, the biotech has infused a fresh energy in the researchers who were actually struggling to get their restriction to cocktail just about right. So I think there's been a tremendous progress on the biologics front. Biotech, I think from the industry point of view, as Dr. Chabria also mentioned, and Dr. Kozo has also mentioned, that the biotech is generating revenue. And in fact, the healthcare industry as such is going through this transition phase where the focus is shifting from the small molecules and getting skewed towards the biotech-based products. There was a time, a few years ago only, there was, I think, the majority of the drugs getting approved or the market share was skewed towards small molecule. More than 70%, even 10 years ago, 70% market share was captured by small molecule, and less than 30% was the biotech-based products. But today, I think we are talking about almost 50-50 market share by biotechs and the small molecule. And thinking of future, next 5 years, I saw one report from consultant that they have projected that in about by 2024, that's about maybe 3 years from now, the biotechnology-based products, the market share will be more than 55%. So they will be dominating the therapeutic product, and they're growing at a very high rate. If you think of last so many years, last few years, CAGR is high single digit or maybe in the double digits. In fact, if you think of top 10 grossing products, I mean, you'll be surprised that today 8 of them are biologics.
Send me the link again, please.
So
Oh, okay.
majority of them
I
are
need to.
biotech based,
Join
and there are only few molecules which are the small molecules reaching the top 10 most selling products. The reason is largely because of emergence of MABs in the oncology space, which is by far the largest segment in therapy, and pembrolizumab, and there are other products which are, single product has captured more than $18-19 billion market, which is probably as much as the entire pharmaceutical market of India is captured by one single monoclonal antibody. And I was looking at the projections, the pembro projection of by 2024-25 is about $24 billion. So I think the future is biologics. So far the spending is also more in the biologics, the R&D, the revenue, as the revenue spend increase, the revenue earning increase, the R&D spending is also increasing in the biotechnology spaces, and there are great opportunities to come up with novel biologics. In fact, biosimilars are also projected to be great opportunities for many companies, and companies are in fact trying to encash this biotech wave. The frontier of innovative therapy is advancing beyond traditional biologics, and when we are looking at gene therapies and cell-based therapies, they are also in action, and these things which are beyond the therapeutic proteins actually are very precise, and they work like magic, So when you identify that the disease is caused by a specific gene or specific mutation, you can use the gene therapy, which are kind of personalized therapies, and they are very effective and they work. And they've been helpful in the growth and development of the biologics as a field, which is coupled by development of application of AI, machine learning, and the appreciation of patient-centric approach. So I think people are more and more focused on delivering magic bullets, magic therapies, which are highly precise. We have seen in the case of cancers that you do diagnosis, find out what kind of drugs are going to work, and then your therapy will definitely work. So those are great scientific advancement to the biologics space. And I think at this time, what better examples to have in front of us than the COVID vaccine? And you'll hear in the next few days, I think one of our Zydus colleague who actually did all the work on developing the COVID vaccine, will be sharing how the Zydus DNA-based COVID vaccine was developed. But I think as far as the technologies are concerned, I think, in my opinion, the scalable and effective technologies are going to thrive in the end. And the pandemic has brought the capabilities of developing nucleic acid-based therapies to a scale and level that they will become relevant for a large population. At one stage, DNA-based products or gene therapy-based products were considered expensive, but we have ventured into developing DNA-based COVID vaccine, which you must have heard is shown great promise in both preclinical and clinical studies, and we are taking it through the phase 3 stage clinical trial now, and there is a possibility that very, very likely possibility that will come to use and get approved very soon, near future. So the possibility with these technologies are immense. We have just begun to test them at a large scale. Now, the success of this kind of products is going to be revealed in time to come. And if that happens, then they are going to change the vaccinology forever, because they are so easy to develop and they are so precise that and so adaptable that one can change them whenever you wish, and they are so stable that you can keep this DNA vaccines at the room temperature without worrying about any changes happening there. So I think what were, in my opinion, what were considered as innovative therapies are going to be commonplace of tomorrow, and I think these technologies are going to be evolved in a manner that they will be very easy. And the only question is how we are able to make them highly scalable and highly affordable. And I think things initiatives that India is taking are in that direction. Now, whether it is making cheap insulin, I think companies like Biocon and Vocard have done great work by producing insulin at such a low price that what was considered recombinant insulin at one stage affordable only to rich people, now every serious diabetic patient can take the insulin shots, and it's really cheap. And a time will come when all these sophisticated biotechnology based products, whether it is a vaccine, DNA based vaccine, or whether it is a monoclonal antibody, or whether it's expensive recombinant therapeutic protein, a complex protein, or the ADC, which are at this moment very expensive and unaffordable to large population, will become accessible to a larger population. And insulin is a great example in front of us. Now, I think while the problem with the scale and accessibility is complex, for I think innovative therapies to become scalable, there is a long road of development. I think it's not easy. So from bench side to bad side, as we always say that, what we do in research is like and the bench of the laboratory to the real meaningful benefit to the patient, that is a bad side. It's not easy to bridge this translational gap, and we call it a valley of death, where most molecules fail to translate into what appear to be very attractive therapy to a very affordable and convenient product for a large population. So eventually we need to develop the biologics. I mean, I think we have to start doing everything right, from the selection of the right primers, development of the stable clone, which are going to produce very high yields, efficient processes, both upstream and downstream, right kind of formulation, and then proper characterization in the form of analytical variables, analytical parameters, all have to be okay. Then only we can have the right kind of biologics. They should get tested in the preclinical models and later on in humans to demonstrate desirable properties. And it's an incremental and systematic approach which everybody follows, but it's prone to failure. So, but we still be trying to develop the vaccines and biologics following this kind of standard battery of studies, and follow the right kind of research approach for making the biologics affordable, and develop them in the right quality and the right price. This kind of work is quite extensive and it's expensive. So, but we are prepared for developing this kind of products in India, and we are well equipped for doing that. developing, I think I'm in the discovery and development of products. So Zydus, as you know, has been developing large number of molecules in the small molecule space, in the large molecule, that's the biotechnology based products. We are also developing next generation of novel biologics, the conventional vaccines, the next generation of vaccines, and also therapeutic vaccines. So development of these drugs, this kind of drugs are always full of challenges. And as Dr. Calder mentioned, whatever can go wrong does go wrong. So I think we are prepared for that. We are prepared for failures. Biologics pose additional challenges because we are developing drugs for humans, and they are, as I was mentioning, these are kind of magic therapeutics. They work very precisely, and they work only in humans. And though, I mean, all of you are scientists, and then to understand while developing the products, we can't directly test the things in the humans, although we wish to, but it's not possible. So the research start from mouse or in vitro to mouse, and then larger animals, and then humans. So many times we face challenges, like the biotech product we are developing does not work in mice. So how will you test it? And if you test it, they have no action. So you have no idea whether it is working or not. So sometimes we have to identify surrogate molecules, surrogate endpoints. We have to use, again, biotechnology to get transgenic animals, which are animals, but they actually behave like humans. They express human proteins or human receptors, and then develop this kind of therapeutics so that we can properly assess the true potential of these molecules. And then eventually we have to test the safety, that these molecules are going to be safe, going to be effective. For biologics, we have additional concern, unlike small molecules, that they have often potential to develop allergic reaction, immunological reactions. So those need to be additionally tested. Specificity is a big issue. It gets evaluated. It takes time to do all these evaluations. Then there are regulatory challenges of developing biologics. I see in your program that you have a talk by Dr. Ramteke, who was a regulator, drug regulator, and he will tell more about the challenges for developing biologics. But for young students, I must tell that it's not easy. There are regulatory guidelines. There are a number of steps that one need to follow. In India, there are new drug and chemical clinical trial rules. For biotechnology-based products, there is a Department of Biotechnology, and within that they have a division called RCGM as a committee, which takes care of all the recommended products, approve give all the permissions for, right from importing the key components to conducting research and various kind of preclinical and clinical studies. Eventually, those studies are conducted and they are evaluated by multiple committees. So you will hear more about the regulatory challenges and the regulatory path from the people who are actually expert in doing that. We have been doing all those things, but I think it's good to hear from the regulators what are the expectations. But everything is possible. Now, what is good is that because of the challenges that are faced because of pandemic, a lot of things have become simplified because of the COVID. Vaccine development was extremely rapid. I think the regulators were very receptive to accelerated approval, and we were able to get it done faster. Right? Now, I think while we do that, there is other thing that people in industry or people in this field, they keep in mind is the competition. There is an increasing pressure. Multiple people, multiple organizations, and multiple companies and countries are developing similar products, and there is an intellectual property which is developed while developing a product, which protects the competitor to develop and launch similar products. So for students, I think they should keep this in mind that while we want to develop any kind of product very rapidly, which is possible because of no technical know-how is available, we are still not able to develop every product because somebody has filed a patent on it and somebody has protected all kind of know-how, the process of developing the product itself and the indications, etc. So it becomes challenging for one to develop a biologics product because the way out through the coming out of patent is really challenging. Unlike small molecule where patent fencing is relatively easy to break, in biologics is really difficult because of the complexity of the biologic molecule. It's not simple to make a copy of biologics. So while we are developing biosimilars, overcoming the patent and still be able to make exact copy is very challenging. In fact, it's very difficult to develop a biosimilar product, although it looks similar, simple that India as a country has become a generic hub of small molecule. And in fact, we are very successful in launching any kind of product. But in terms of biologics, it's really challenging to come with the biosimilars and demonstrate that it's exactly same as what was innovator's product, because innovator product itself keeps changing. What they develop in the beginning, what they are selling now is actually changed because of the number of factors that are involved. So coping up with those kind of challenges is really difficult, but we have still find a way out. People also try to protect their biologics by coming with the follow-on products, that the next generation biologics or something which is a little better than what they were producing earlier. Although it becomes commercially challenging to sell the follow-on biologics, but there are opportunities for one to develop follow-on biologics. And novel biologics is another thing that one can think of developing, where you don't have to compare your products with the other products in the market. So now a lot of companies are developing novel biologics. There's also a perceived threat to biologics from the small molecules. I think at one stage we thought these are two separate streams where biologics will have their own targets and their therapeutic domains, and small molecules are having different druggable indications. But now I think people are becoming smarter. There's a lot of progress in the small molecule space, and they are actually posing threat to biologics. For example, I will give our own example. We are developing a small molecule Hpv stabilizer that will be used for treatment of anemia, which is currently treated by erythropoietins. So once we have a small molecule developed, erythropoietin will become not so attractive because it's injectable, and whereas small molecules will be taken as oral tablets. As far as cost is concerned, small molecules are always cheaper than the biologics. So these are kind of perceived threats to biologics. So one who is keen to think of a novel biologics, one need to think of potential threat that can come from the other competition from not only biologics but also from the small molecules. I think these are some of the challenges of developing biologics as we understand. These are when I was talking so far, I was talking of therapeutic proteins and monoclonal antibodies, which is what most Indian companies are currently developing and producing. But biologics space has evolved beyond therapeutic proteins and MABs. And I was talking about earlier, I think the ADCs are also attractive option where you can combine the small molecule and biologics by chemically conjugating them to give new properties, which are a very attractive segment at this time. And there are already few products approved, like Kadcyla, which is a mAb which is conjugated to a toxin which delivers the toxin to the cancer cell and then is very effective. So there are targeted therapies, and there are opportunities for one to think of next generation of therapeutics, and I think some of these are actually developed by small biologics companies, startups, academic institutions. And I think LMCP, AIC kind of incubation facility, something is a nice setup that you have where initial research could be done, which could be acquired by a large company to take the concept forward. An example, the Spark Therapeutics, which produced finally what became Luxturna, was acquired by Roche by paying a lot of money. And it's a kind of a novel gene therapy for correcting a form of genetic blindness. I think that is true innovation and true disruption, that you can use a gene therapy for treating a serious diseases. So what can find own niche, and there is opportunity for institutions and small biotech setups to develop a innovative kind of therapies. Then there are, I think, a lot of other things which are, I would say, getting into a different kind of domain. Where if I talk of this LMCP is organizing this symposium, I want to talk about challenges that are faced in delivering the biologics. And I think the major challenge is the route of administration. Most therapeutic proteins and biologics are parenterally administered. And although some companies try to develop nasal delivery of therapeutic proteins, but eventually they did not succeed. So there is an opportunity for institutions like LMCP and scientists who are in this field, pharmaceutical field, to complement the biotechnology product development by developing novel novel formulations, which can either change the way that this product could be administered, modulate the properties to some extent so that they become more effective, or more stable, or could be produced faster. Some of the process improvement, like better lyophilization, fast lipho cycles, better yield, more stability at room temperature. These are some of the avenues that are there for pharmaceutical scientists to complement the biologic protein or therapeutics development. There are also, I think, opportunities to develop or take this thing to a next level. Apart from treating the infectious diseases, there are a lot of therapeutic biologics now available. For example, anti-PCSK9 antibodies, they became a kind of disruption for treating the hypercholesterolemia. And most recently, yes, late last year, first in class a gene therapy was approved, which is a siRNA based platform to neutralize PCSK9, also got approved by FDA, where you can really neutralize the protein in the body. The body does not produce the PCSK9, and thereby you achieve the same kind of output as you achieve by anti-PCSK9 antibodies. So these are, I think, some of the next level of advancement in the biotech space. Then another thing that is futuristic. Actually, I'm talking a little bit about future because we are beginning a new decade, and I think, as our Prime Minister Modi also mentioned, that it's the beginning of decade. So whatever is the future is, we should see the beginning of that in this decade. So, and this is a very appropriate time. That's the beginning of the year, the beginning of the decade, where we are talking about what is going to be the future of the biologics. So I mentioned about this siRNA approach. I also, when a few years back, we organized a symposium called Raman Foundation Symposium, and we actually are very fortunate that we know the person who discovered or developed the humalin, the first recombinant insulin, is Dr. Richard DiMarchi. He is closely associated with us, and we invited him to give a talk on what he thinks is the future of biologics. In fact, he was sharing the new kind of biotechnology advancement that he and his collaborator Pete Schultz developed at a small biotech company in California. What they did is really interesting. We all know that we have 20 amino acids. In our body, that, and they create all kind of proteins. It's just the jugglery of this amino acid that gives rise to different kind of proteins, right? Now, I also talked about the next generation of biologics that we modify the protein to attach something like either toxin, or people attach PEG to create a longer acting proteins. But the problem with this chemical conjugation is that this other moiety can get attached to anywhere, because the proteins are very complex. Their three-dimensional structure is very, very, very complex. And if it attaches something to, for example, alanine or cysteine, and then there are multiple cysteine, your molecule get attached to any of the cysteine. How do you control where you want to attach? Now, if you're not able to control, you can't control the quality of the product. So the solution came from the pioneering research done by Dr. Richard DiMarchi and Dr. Schultz, is that they introduced an unnatural amino acid. They designed the bugs that actually produce or introduce a unnatural amino acid in the protein. At the place of their choice, they can decide and choose that I want to have this acetylated alanine at a specific point. And by introducing it at this point, the bacteria, the E. coli, can produce the same protein with this artificial amino acid introduced and produce it at a large scale. Once this protein is produced, you can run a chemistry, and then you can modify this particular amino acid to produce a product of your choice. So this opens up a plethora of the opportunities. You can create designer biotech products. I think it will be a matter of time that those kind of technologies where, in fact, I was listening to Pichos later on, and then he has introduced multiple novel unnatural amino acids that can be produced by the E. coli. So we are actually playing with the nature. We are going beyond what God has given us to create a battery of amino acids that the bacteria are fooled, and they're producing it very efficiently. So that's the future of the kind of biotechnology. The other thing that I consider as a future, India has been a generic hub. We have been producing small molecules, APIs, and generic drugs. Many times the generic drugs are difficult to produce, but now people have started using biotechnology, and this is something that is called as chemical biotechnology, where the molecules, which are actually very complex, can be produced by bacteria. So you can design the bacteria to produce alkaloids, for example. Now we know alkaloids are found in nature, but can you, for example, certain plants produce morphine? I mean, it's a banned substance, but it's produced by opium, right? But can you have a E. coli produce morphine? People have demonstrated it's possible. You can design, not E. coli, but actually they did it with the yeast. So they made yeast, which are engineered yeast to produce large number of alkaloids. I think that, I believe, is the kind of future of producing complex chemical entities. I also talked about future where people can change or introduce novel substituted amino acid in the therapeutic proteins and create drugs. I think the opportunities are many. India as a country, we are at this moment, we are excellent in making generic drugs. We have set example that we can make new drugs. We have built a lot of capabilities of developing biosimilars, as you heard from previous speakers, that large number of companies are actually in the biosimilar space, and we have good capability. And few companies have also entered into a novel biologic space. And Zydus I'm very happy that we actually launched our first novel biologic, that's a rabies monoclonal antibodies. And we are working on a large number of novel biologics. I think the future is very bright for a country like us. We need to spend a little more money on the research and development. And I'm very encouraged that this year's budget has allocated more research funds for research and development in healthcare. And I believe that the government labs will definitely get an advantage of that to create basic fundamental research and identify some commercially attractive technologies that could be taken up by industries. So I think these were my thoughts, and I think this is a great talk. We need to move from the brownfield project, as we say in industry. That is something that is already known. We are trying to make an incremental, a greenfield innovation project, which is right from the starting, something from the concept stage. And I wish all the young researchers all the best. I hope that some of them will be very creative and come with the really innovative products that could be taken up further. I also wish that many industries come forward to do research and produce the novel biologics and the biosimilars that will change our landscape, and we truly become the frontier country in the biologics space. I want to congratulate the institute and the organizers for organizing this very interesting meeting, and I'm happy that I was able to share some of my thoughts and views to all of you. And I wish all the best to for the conference. I hope that this is very successful. In the next few days, people get to learn a lot more in details. Thank you very much.
Thank you, sir, for gracing this occasion and giving us an insightful speech and in-depth knowledge on the upcoming opportunities in the field of biologics and biosimilar. Now I request our principal, sir, to present a certificate of appreciation to Mukul, sir, for sparing his valuable time from his busy schedule and accepting our invitation. So I request Sandeep, sir, to share his screen so that principal, sir, can present the certificate virtually. Sir, please accept this. And thank you very much.
That's innovative. Thank you very much. I receive it.
Okay, sir. So now, coming to the end of this inaugural function, I now like to invite Dr. Praful Bagadia, sir, on this platform for giving vote of thanks. Sir is professor in the Department of Pharmaceutics and Pharmaceutical Technology at LM College of Pharmacy. He has around 30 years of work experience. He has guided around 11 PhD scholars and has filed 9 patents, out of which 2 have been granted. Talking about his publications, he has around 103 research publications and 5 books published. He is recipient of various awards like Indian Achievers Award for Education Excellence in 2010, Rashtriya Gaurav Award in 2010, and Indira Gandhi Siksha Shiromani Award in 2013 for outstanding achievements in the field of education. He is also member of various organizations, to name a few, like IPA, ISCE, APTI. Also, he is SSIP coordinator and member Board of Studies, Gujarat Technological University. His current area of interest is modified excipients, cGMP implementation, parenterals, pharmaceutical process engineering. With this, I invite you again, sir, for delivering vote of thanks.
Thank you very much, Dr. Radhika. Hello, everyone. Respected members on the virtual dais today, Dr. A.H. Kalro, B.M. Shah, Principal Dr. M.T. Shabaria, Chief Guest Dr. Mukul Jain, Dr. Yamini Shah, and the anchors over here. It is my proud privilege to present the vote of thanks for this wonderful inaugural function of the International Conference on Biologics in Healthcare 2021, Unfolding Genes to Proteins. I present a vote of thanks. We are very grateful to the Ahmedabad Education Society, the management of LM College of Pharmacy, for providing us the opportunity and supporting us to execute and have this program. We are indebted to Dr. H. Kalro and Shri B.M. Shah for being the patrons for this conference. We are also thankful to the sponsor of the conference, Gujarat State Biotechnology Mission, and our industry partners, Thermo Fisher. I thank Principal Sir Dr. M.T. Chabria, the convener of the conference, for highlighting the biotech industry in India vis-a-vis what is happening in the world today. I thank Dr. Yamini Shah, the organizing secretary, for giving an overview of the conference schedule, which is going to be held during these 3-4 days of the exciting lectures from various eminent speakers across the globe. I thank the chief guest, Dr. Mukul Jain sir, for providing an in-depth idea about so many things which are going on in the field of biologics these days. It reminds me of the genome, that's the human genome project, which started in 1990, because it ended in 2003, and I was so excited that we started to establish one of the first biotech labs in one of the institutes, and we had an investment of about 1 crore which was made in that lab at the end of the establishment. So that is, the excitement which comes in when something new is happening. So we thank Dr. Mukul Jain sir for his in-depth ideas about how the challenges and threats are there, how we are shifting from small molecules to large molecules, and the opportunities coming up. Thank you so much for sparing your valuable time and being with us here today. We are also indebted to our guest of honor today, Dr. A.H. Kalro sir, who is the academic advisor at AES. Sir, we are always waiting to hear the words of wisdom from you. You have always been very inspiring to us, and your points on focus on education, focus on entrepreneurship, research and innovation have always been at the forefront. We really understand this, and we do ensure that we will try to focus on these issues. And we also, yes, appreciate your views that unbelievable advances in medical and healthcare sciences are taking place, and people from our country have contributed a lot to these advancements across the globe. Thank you so much, sir, for sparing your time and being with us today. I also thank all the committee members and for putting so much of efforts in organizing this conference, the participants, fellow colleagues, friends, students, not to forget all the learned speakers who are going to address the participants of the conference, the chairpersons, the panelists from across the globe. So with these few words, I'll be very short because we are getting delayed because of a small technical glitch, and therefore I wish all the participants a very enthralling experience in these 4 days of the conference. I thank all the invited guests here today for being with us today and gracing this inaugural function. Thank you very much. And I hand over the mic to Dr. Radhika again.
Thank you, sir. Thank you very much. Now I think we are ending our inaugural session over here, and we will now soon begin with the first session of this conference.
Thank you, sir.
Thank you, sir. I thank
Thank you so much, sir.
all the
Thank you, Jen, sir.
attendees on this virtual
Thank you.
platform
Thank you,
for
Radhika,
being
for
here. Thank you, sir. So now, without delay, I move to our first session. So before that, I have some few announcements. So any questions which are raised during the session, it can be sent through the chat box and it will be answered at the end of the lecture. Also, a link will be provided for the attendance, quiz and feedback at the end of all sessions each day, which is to be filled compulsorily by the participants for availing the certificate of attendance. Now, we will start with our first technical session. And for that, we have with us our first chairperson, that is Dr. Vivek Upasni, sir. I think sir is joining in few minutes. Ketan sir, Vivek sir is joining.
Yes, madam, he is joining.
I guess Hemant sir has joined.
Yes, Dr. Hemant is with us. We are waiting for Dr. Shardul and Dr. Vivek to join us in the room.
Hello.
Hello. Shardul sir, sir, we are waiting for Vivek sir to join.
Sure.
Sorry, I think Vivek sir has some joining issues. So I will introduce our first speaker of the session, Dr. Shardul Salunkhe sir. I welcome you, sir, over here. Vivek sir will join us in short time. Yes, sir. Because of paucity of time, I will introduce you. So, sir has completed his graduation in veterinary sciences and PhD in biotechnology. Sir has done post PhD. Sir has joined Lupin Biotech and contributed in R&D for the production of the biosimilar molecules. In 2017, he shifted to Intas Biopharm as AGM and head clone development team. He has contributed for the development of high titer clones for Intas Biopharm in quick time and has also worked in the early stages process development. He has huge experience in biosimilar molecule development for Indian as well as regulated market like EU and US. So with this, I welcome sir. And sir is presently AGM clone development in Intas Biopharma, and he will be delivering talk on basics of clone and cell line development for biosimilar project. So sir, I welcome you on this platform. So you can start your session.
Thank you. Shall I share the presentation?
Yes, sir, you can share.
Okay. It's sharing. It is sharing.
Sir, you are sharing screen or you are uploading the slide?
Okay, I'll upload the slide. Just a minute.
Or sir, you can share the screen, the third icon on the topmost part of the screen.
Okay.
Besides audio, we have a screen sharing button.
All right, I will open the presentation and then share.
Okay, sir.
Okay, can you see my presentation?
Yes, sir. We can see it clearly.
Okay. Good evening all to all the audience first. And secondly, thank you to the organizers of this conference for inviting you for the talk. So first of all, the name of the conference is Biologics in Healthcare 2021, Unfolding of Genes to Proteins. So the role of my department or myself comes in between. So from taking out the genes, putting it into the different cells, either E. coli or CHO cells, mammalian cells, and then expressing those genes into or converting those genes into the proteins. So that is basically the work of clone development or cell line development. So when it's in E. coli, we call it clone development, and it's in mammalian cell, we'll call it as a cell line development. So I will go ahead with my presentation. I'll start it. This is in short what I will be explaining. I will be first talking about few points about Intas Pharma, then some pointers towards what is biologics and biopharma industries, followed by I will be explaining what is exactly recombinant DNA technology and how the cell line development process takes place. And then if time permits, I will also explain some parts of the monoclonality and why it is required. So Coming to the second part. So firstly, talking about Intas Pharma, Intas started as a company, as a firm in 2000, year 2000, where immediately after 4 years of its start, it launched its first molecule, that is GCSF molecule, which is used in neutropenia. Then after that, subsequently, every second or every third year, it has launched molecule in Indian market. The milestone is where in 2015, Intas launched filgrastim in Europe and Canada, and in 2018, it launched a pegylated version of GCSF in Europe and Canada. So those are the major milestones for Intas Biopharma. And currently, Intas Biopharma is one of the top companies in India, and also one of the big companies in the world. So that's about Intas Biopharma. Just going through the basics, since I am assuming that most of my audience is from the pharma background. So to compare what is the difference between small molecule and the protein drugs which we work upon. In the first slide, there is an aspirin molecule which has only 21 atoms, as compared with human growth hormone, which has about 3,000 atoms. So that is the difference between the small molecule drug and a large molecule drug. And then finally, there are even larger drugs like IgG antibodies, which we work on. So those are even bigger. Those range in size of 150 kilodaltons. So that is a quite huge molecule to work on. So there are multiple physicochemical properties, biological properties, which vary from each molecule to molecule, and that we have to study deeply. So the complexity can be understood from the lower panel, where one side it is a simple cycle, and at the other end it is a business jet which weighs around 30,000 pounds, has multiple functionalities to operate that jet. So that is the level of complexity which I want our audience to understand for the comparison between small molecule to the protein drugs. Then coming to what exactly are the currently top 10 selling drugs in world over. So as we can see, these are the top 10 drugs, out of which only 3 are from the non-biologists, while the rest of the 7 are all produced by biopharma industry. So that shows that the future of our biopharma industry is directed towards biotechnology mainly. And even in future, there will be a lot of molecules which will be coming and are in different stages of trials, clinical trials, and phase 1, phase 3 clinical trials. So, just to explain what are exactly biosimilar molecules. So for example, our first top drug is Humira, and the chemical name is adalimumab. So whenever the patent for adalimumab gets expired, we will launch a copy of this molecule, copy of this adalimumab molecule. So that is called as a biosimilar molecule for the adalimumab. So that is which mainly Indian companies focus on development of biosimilar molecules. And the Intas is also focused on biosimilar molecule development. So since I wanted to start from very basics of clone development. So first I will explain which most of you might have known and might have read in your biology books, is the central dogma of life. Always the first starting point is the DNA. DNA gets transcribed into the RNA molecule, and then the RNA molecule gets transcribed into the protein molecule. So this is the exactly same thing which we do in our laboratories. This is done naturally by cells routinely, and this is what we force our cells to do and produce our interest of protein. So the DNA will be cloned. DNA, which is gene of our interest, will be cloned in the expression vector. It will be introduced into the cells, and then it gets transformed, and then it expresses out protein. So this is what the basis of our life, and this is what is the basis of our biotechnology industry, which produces the biologics. So how exactly the biosimilar drug gets development? What are the different departments or different functionalities that are involved in the development of biosimilar drug? So always the first department comes is the clone development or the cell line development department, wherein, as I explained, we have to extract the DNA from the natural source or the artificial source and clone it into the plasmid. This circle here shows the plasmid where the gene, our gene will get cloned. And this plasmid will then either get introduced into the bacteria or a mammalian cell as shown here. So once the DNA gets introduced, so it is called as a clone. Yeah, so I have a pointer now. So once it gets into clone, once the gene gets introduced into the vector, it is called as a clone. And then we confirm whether the clone is having right amount of expression, right amount of the quality of the protein. Once that confirmation is done, then the cell bank is prepared, which is a source from where your next batches will start. So then followed by clone development, next department is the upstream department, where protein production takes place, where we cultivate this either the mammalian cell or the bacterial cell in huge volumes, in like 1000 liter, 2000 liter volumes, followed by the downstream department. So the downstream department, the main function is to purify the protein, and we use multiple chromatographic columns to purify the protein, remove all the impurities, and your drug should be 99% pure. And then the final product is formulated into a formulation where it will be stable, and then this molecule will initially will go through the different preclinical and clinical trials, and when everything gets approved, then the molecule is launched in the market and will reach to the patient. So that is the in nutshell what is exactly a development of biosimilar molecules. Now coming to recombinant DNA technology and coming to the basics of the clone development. Most of you might be knowing that double helical structure of the DNA was discovered by Watson and Crick. But there are two other people other than Watson and Crick who also needs to be mentioned here, are basically the Wilkins, who was an X-ray crystallography expert, and then Rosalind Franklin, who was a lady who was working in his lab, Wilkins lab. Both of these scientists actually developed this X-ray crystallography image, which gave a clue to Watson and Crick. And then Watson and Crick could develop the three-dimensional model of the double helical structure. And from this, actually, DNA technology exploded, and many more technologies were developed from this. So all the three scientists were awarded Nobel Prize for this particular work. So how was the early technology developed after the discovery of the DNA structure? So there is a group of patents, also known as Axel patents, which were filed in 1980s. So this Axel is basically a group of three scientists. They were working in Columbia University, and they basically patented this technology wherein you can code a DNA in one plasmid and code a selectable marker in another plasmid, and then both of them can be co-transformed to prepare a clone. So this technology was first developed by them. And then all the big firms which we hear, like Amgen, Biogen, Genentech, all these firms purchased this patent from the developer from the Columbia University. And later, even 24 more other firms also purchased. So now, currently, this Columbia University earns close to $790 million of royalty every year. So that is like a achievement for our science, our biotechnology industry. Once the technology moved from university, it was directly replicated inside a biopharma industry. And so first molecule that was launched, that was Genentech. So Genentech was the first company which launched first molecule. It was a factor VIII molecule, and it was launched in 1987. So this opened an entirely new department where drugs can be produced. So that is biopharma for us. So coming to the actual general cell line development process. So here again, there is an expression vector where our gene of interest gets cloned, and then we mix this expression vector containing gene in with the host cell. So, and that process is called as transfection, where we introduce our DNA into the host, and followed by that, our plasmid usually contains a selectionable marker. Usually, it's an antibiotic, or it can be also any other genes like DHFR gene or GS gene. So these help us in selection. So out of the pool where the transfected molecules will be present, as well as the non-transfected molecules will be present, only the cells which contain the expression vector will get selected, and rest all the cells will die. Then we go to next step, which is called as single cell cloning or limit dilution. So in this case, what happens is, since our pool contains mixture of cells, some cells will give very low expression, some cells will give very high expression. So what we do is we plate individual cell in each of the well in 96 well plate, and then screen for better producers, screen the cells for better titer, so that we can get higher and higher production in the bioreactors later. And this can be repeated multiple times. And then followed by this is taken to the next step, where we expand these clones from 96 well plate to different levels. Then finally they reach the shake flask. At the shake flask, we test again for top 10 clones for like expression and the quality. And then, as I said earlier, the cell banking is prepared. And then once the cell bank is prepared, this will go for further evaluation. So that is the process for a general cell line development, which is used across the world. So usually, what is the ideal clone, or what is the ideal cell line that we are targeting? So firstly, it should be stable. So what do we mean by stable is that it should be scalable. Like if your highest scale is in say 1000 liters or 10,000 liters, our clone should be stable throughout the passages and express the same amount of protein which it had produced in shake flask or smaller scale. So that's why it should be stable. Then it should be monoclonal. It should be coming from a single cell. So that is very important. Otherwise your clone will show a variability. It might express sometimes, it might not express, or it will show variation in the quality. And then third point is very obvious, that it should be producing very high titers. Like currently in Indian companies, it's mostly in the range of 2 to 4 grams per liter, which is considered good titers or high titers. While in foreign companies or the publications, it goes even beyond 5, or even it reaches to 8 to 9 grams per liter. So that is the range which we see. Then when we are working with biosimilar, since we are copying the innovator molecule, it's very important that our quality should be very, very similar to that of the innovator molecule. Whatever your physical characteristics, chemical characteristics, biological characteristics, all characteristics should be matching to the innovator. Then only our product will get approved. Otherwise, it will not get approved. And finally, the clone should be scalable. It should be scalable to the very high scales. So these are the parameters where we look and study our developed clones and select the best clones available. So coming to the technologies that are available currently in market. So there are different companies, as I mentioned here: Celexis, Merck, Millipore, Horizon, Lonza, Thermo. So all of these companies sell their own technology for the production of biosimilars or biologics. But the negative factor of this is that we have to pay them royalty. We have to pay them license fee, as well as the upfront fees, and these fees are huge, in some $1,000 or some $1 million. So another thing is that we can develop these technologies in-house. So we basically will develop our own vector and we'll work on that. So I've just tried to explain here. If you can see this image, so this part of the genome is called as heterochromatin, and this part is called as euchromatin. So when the DNA structure is very much tightly packed, the kilometers and kilometers of length of DNA is packed inside a tiny nucleus. So that packing is called as heterochromatin. So as you can see here, whenever this is RNA polymerase which converts our DNA into protein, DNA into RNA. So so if there is no space to RNA polymerase to bind, it will not express itself. So it needs to be converted into euchromatin. So these are the euchoi, as I have written here. These are particular gene elements that are incorporated into your vector so that your vector gets opened and RNA polymerase can bind and produce RNA, and then in turn it will lead into production of protein. So these type of technologies are available in market, which can help us in production of getting higher titer and getting higher producer clones. So this slide shows what exactly we do in Intas Cell and Development Department. So usually our process starts from molecular biology, where our gene gets cloned into the expression vector, followed by, as I said earlier, transfection, and then after 1 week we'll go for plating the transfected pool into different 96 well plates, and usually it's around 5,000 cells per well. So this is called as a mini pool generation. Followed by we have one step mega pool generation, where we pool our top 10 mini pools, and then we go for single cell cloning using limiting dilution. So that is a process where, as I said earlier, where we do a single cell cloning, and these cells are imaged by an imaging instrument called as single cell imager. So from this, again, once we after 14 days, we'll get a group of colonies. We'll screen them for production. We'll expand them. This takes around 5 weeks, and we'll get a stage where we can select top 30 clones, which can be either analyzed in a shake flask or in a mini bioreactors like amber, which I have shown here in the image. So this amber can be used for like 48 clones can be studied at a time. Somebody has to mute the mic.
I think, yeah, everybody please mute yourself.
Yeah. So from these top 30 clones, we come to the top 10 clones, which go under quality analysis. I will explain what exactly quality analysis we do in next slide. We do a reproducibility analysis of top 10 clones, and then finally our research cell bank, or the cell bank, is prepared for the top 5 to 6 clones, and these clones get transferred into the next department, which is Upstream Department for process development. So this is a nutshell what we do in Intas. And as you can see here, the process takes usually 5 to 6 months in total to get from transfection to the clone development. So coming to the next, what exactly quality parameters we look for biosimilar projects. So as I said earlier, biosimilar clones, biosimilar protein should be exactly similar to the innovator molecule. So what we do is we do a battery of tests during for our top clones, which contains like mass spec studies, peptide mapping, then different impurities are studied. Even impurities are also important in development of biosimilars. Your impurities also should be very low, and it should be somewhat matching to the innovator impurities. So for impurity analysis, purity analysis, we do SCHPLC, RPHPLC, then CESDS studies. And there are one of the important quality parameters is the glycosylation of our protein molecule. So this also we do at different levels, either at minipool level or at the clone level. So basically, we have to study the RNP molecule. We have to go into deeper, and then we have to study our own molecule and compare it with the innovator, and test whether our clone is correct or not correct, and how much our clone is closer to the RNP. So those, and if there are any additional impurities, we have to do mass analysis and characterize them. So that is another study which is required to be done. So apart from actual clone development, there are other clone related activities which we are required to do So we have to do sequencing, RNA sequencing, then cDNA sequencing, where we check whether our DNA sequence is correct and matching to the innovator. Then, as I said, we do clone stability experiments, so that we do testing for 70 to 80 generations, and we'll test whether our clone will withstand the passaging till 1 kL or 2 kL bioreactor batches. Then we have to do cell bank development, like master cell bank, working cell bank, all those banks we have to develop and then get them characterized. And also some part of preliminary upstream experiments are to be done. So these are additional clone related activities apart from the pure clone development that is required to be done. Do we have more time? I can go through 5-6 slides for monoclonality.
Yes, sir, you can go ahead.
Sure, thank you. So as I described earlier, so monoclonality means that our clones should be produced from single cell, only one cell. So that is very important. And why monoclonality is required? So all the regulators across the European market or US market, so what their guidelines clearly specify, like in ICH Q5D, they say the cell substrate has been cloned from a single cell progenitor. Then another EMA guideline also says that it should be coming from stable and continuous monoclonal cell line. So this is a regulatory basis, and what is the scientific basis is that if our clone does not come from single cell and it comes from the multiple cell, so there will be heterogeneity while preparation of master cell bank, and its characterization will be tough. As well as when our cell banks are non-clonal, there is a chance that changes can be made or changes can happen during upstream process, so that can lead to change in our titer, change in our quality. So that's why monoclonality is important. So there are different methods that can be used for this monoclonality achievement. So that can be done like one method is single cell cloning. So this can be done either manually, or we have FACS sorter machines, or we have cell seeding equipments that can be procured and that can be used for this seeding of the single cell in a well. And also there is an instrument called as ClonePix, so which helps us in selection of cells, which can be selected based on fluorescence. So this instrument can also be used for single cell clone purpose. Only drawback is that if we do ClonePix, then we have to do 2 rounds of cloning to confirm the monoclonality, which might increase the timeline, which will increase the timeline. While in case of single cell cloning using lipid dilution method, it will be done in much shorter time because only 1 round of cloning is required. There are like multiple instruments which are to be used for imaging of the cell. So I have mentioned here few equipments that are available in market, like Cell Matrix CLD, Clone Select Imager, IncuCytZine, and there are different seeding instruments which help us in seeding of these cells that are available, like WEBS and single cell printer. These are available in market. This helps. So these instruments have already been procured by INTAS to ensure the monoclonality. So whatever the clone which comes out of our INTAS clone development department, it will be 100% ensured that it is a monoclonal, and it will not have any issues during the scale up and production process development. So I want to show one of images, how the images are developed. So this is a well from a 96 well plate, which we can see here, and this is an image of that well taken at day 0. Day 0, and this is a zoomed image of this well. So as we can see, you can see here a single cell, only a single cell in the well. So this ensures that our clone has come from single cell. And then we go to day 1. So we can see the one cell has now multiplied and converted itself into double cell. Then going further, this 2 cell has developed into a small colony, a bigger colony, and then entire plate gets covered in this colony. So as you can see, you can trace back that your clone or your colony has come back from a single cell. So this is this is how we develop our clones, so that they are best quality and they are good quality, and they can be at par with anywhere in the world in terms of quality as well as in terms of titer. So thank you, and if any questions, they are welcome.
There are a couple of questions. One person is asking, what are different types of vectors which we can use?
So vectors, types of vectors means there are mainly in biosimilar industry, we don't use viral vectors. Mainly we used plasmid based vectors because they are easier to go through regulatory approval. So basically plasmid based vectors are mainly used, which contain like selection marker and then expression elements like promoter, terminator, origin of replication. So mainly plasmid-based vectors are used. Have I answered you the question? Yeah.
Yes, sir. Sir, one Yashika is asking, do biosimilar work in the same way as their biological reference product?
Yes, absolutely. They are expected to work exactly same as the innovator molecule, because doctors are going to eventually replace our biosimilar molecule in the place of innovator molecule. The main advantage is like if the like for example, ranibizumab drug or other drugs cost in range of 70,000 to 1 lakh rupees for an innovator molecule, but our molecule will be available at much more lesser rates like 20,000 or 30,000 rupees. So that will definitely help our Indian patients. But we don't. It's not like we are going. We will be giving something which doesn't work. Absolutely, it will work, and it will work 99% similar to the innovator molecule. Yes.
And sir, I think one last question. In
Sure.
cancer therapy, nowadays monoclonal antibody is used as a target drug delivery for herbal and allopathic drugs. Is there possible monoclonal antibodies coating?
I didn't understand the question actually, but
Sir, I guess means I assume that it is like we can we coat, like we coat a tablet, can we coat monoclonal antibodies? I guess like this is someone is asking.
so usually when the question says that can the molecular antibodies are used in combination with toxins, so those are called as ADCs, antibody drug conjugate molecules. So wherein our antibody will target a particular cell, and then toxin will go there and kill that particular cancer cell or any other cell. So that is what that is next level of development. So currently, Indian companies are more focused on only monoclonal antibodies, but in future we'll be going ahead with ADCs also, antibody drug conjugate molecule.
Okay, so thank you very much, sir, for your excellent talk. We appreciate
Thank
that
you.
you spare your time, valuable time, and I know the session is somewhat delayed, but thank you very much, sir, for joining us and giving us an overview about what is the clone, how to develop a clone, and the related aspects.
Thank you. Thank you. Anybody wants to reach other outside, I am available on LinkedIn. Thank you, everybody.
Okay, okay. So participants were asking about how to contact you. So sir is telling that all of you can contact him through LinkedIn. Thank you, sir.
Thank you. Bye.
Bye, sir. Uh, sir, you can just close the browser. I think Sarfraz sir has joined us. Dhaval sir is joining.
Yes, I'm here.
Sarfaraz sir, Dhaval sir has joined. I think he has not joined yet. So we have with us Sarfaraz sir. Due to some busy schedule, the second speaker talk is, I think, delayed. So we will be right now starting our third session by Professor Sarfaraz Nanomedicine. Sir,
Hello, can you see me and hear me?
sir, I can, yes, we can see you very well and hear you also.
But the screen shows a lot of multiple screens. Is that how supposed to be?
Sir, are you going to screen share? Do the screen sharing?
Is my screen okay right now?
No, sir. Multiple screen is visible.
Showing, huh? Okay, very
Yes.
good. One second.
Sir, do it once again, I guess.
How about now?
Sir, now we can see you.
Okay. And how about my screen?
Sir, we can't see. Now it is coming. Yes, we can see
Okay.
the screen.
All right. I think we all set.
Yes, sir. But so before we start our session, I would like to introduce sir. Sir is a globally recognized. Sir, just a minute. I think there are some joining issues with our chairperson. So, sir is a globally recognized pioneer of making life-saving drugs accessible across the globe. According to Forbes magazine, the most interesting man revolutionizing the health world. Sir is making biological drugs, biosimilars, gene vaccines, gene therapy, cell therapy, and gene editing accessible to the developing countries. I think Dhaval sir has joined. Sorry, sir.
Okay, all right. Yeah, I saw them. Yeah.
Dhaval sir, are you there? Dhaval sir, can you hear me? Hello, sir. Dhaval sir. I think there is issue. Some issues are there. He has joined. His name is there, but
Yeah.
not responding.
No, take your time. No rush. I hope people are patient enough to wait for us. There are 180 people.
Hi, I can hear you. I don't know whether you can hear me or not.
Sir, we can hear you. Dhaval sir, we can hear you.
Okay, fine. Thank you.
Okay, sir. So, sir, now I would like to first introduce you. So,
Yeah, please go ahead. I mean,
yes,
you
sir.
want.
Yes, sir. Just a minute, sir. So I would now like to invite our chairperson of this session, Mr. Dhaval Trivedi. Sir is working as AGM Strategic Planning and Operational Excellence at Syngene International Limited. Sir is an MTech from ICT Mumbai. In his career of over 16 years in biopharmaceutical industry, he has worked at various positions in the work areas of process development, regulatory affairs, and medical device development at various organizations like Intas, Biocon, USV, and Stellis Biopharm. He played a key role in the development and commercialization of biosimilar products for global markets in multiple organizations. With this, I welcome you, sir, in this virtual platform and request you to take over the session and introduce our speaker of the day, Professor Sarfaraz Nanomedicine.
Thank you. Thanks for the nice introduction. And I wish everyone who are on the call to have a good day. I mean, we can see that people are participating across the globe. We have like 180 plus attendance, and that's really fantastic in this time. And it's a beauty of a virtual world that made it feasible to have global participation. So actually people can we have like 180 plus attendance, and that's really fantastic in this. I think there is some echo. I don't know whoever has joined, please. Okay, thank you. So thanks. I mean, I would like to thank Dr. Chabaria, Dr. Yamini, Vivek, and the whole organizing committee to give me the opportunity to work as a chairperson for this exciting session. We all are going through a tough times, but equally exciting times where vaccines are developed and launched in just over a year, which usually takes like more than 10 years to develop. And it's really a breakthrough event. And needless to say that the first ever candidates of vaccines which are launched was with a partnership of an academia and industry. So it came from Oxford University, and then it was tied up with an industry. So this is clearly an example of an industry-academia partnership and importance of academia and its direct contributions. And this is one more event where we are seeing a lot of participation from the industry and academia together, which is going to be helpful for further development. So this COVID is going to change, many things, and let's see how we can use it for the best out of it. We have seen many breakthrough things, and what is the better time to hear from Dr. Niazi as a speaker for this session to understand what are the exciting time and what going to bring the biology is going to bring to the world. So, I would like to quickly introduce him. I think many of them who are actually involved from industry or who are working in regulatory, there will be hardly anyone who might not be aware of who Professor Niazi is. But just a quick words around his introduction. He is a globally recognized pioneer of making life-saving drugs accessible across the globe. He is also a person recognized by Forbes as the most interesting man revolutionizing the health world. His areas of work includes biological drugs, biosimilars, gene vaccines, gene therapy, cell therapy, and gene editing accessible to developing countries. He is also an expert in US FDA. He also working as an advisor to various regulatory agencies. He was part of an advisory group set up by Obama. He is also a researcher with highest number of solo patents, licensed patents. He is also a licensed patent practitioner, visiting professor in the Illinois University. Many times I surprise how can one person can wear so many hats at the same time. Despite this, even if we look at this is not it. He's also wrote dozens of books. He's a keen interest in philosophy. And write poetry. He reached to over 1 billion people through TV programs related to poetry as well as on the related things, including the philosophy. And he's also reachable as a consultant through his own consultancy, Pharma Science. Rather to wait anything more, I really would like Dr. Niazi to take forward this session related to shift of biological research and future prospects. Welcome, Dr. Niazi, on the session, and I hand over to you.
Thank you so much, and thank you for this kind introduction. Definitely, these are the times, but my talk today is about a very different topic. Where would this COVID take us? This was a classical, what you call paradigm shift. Okay, so I'm very happy, first of all, to be invited to this conference and to have so many people. I look for India as the leader for the future of all pharmaceuticals. So I'm presenting today a few things which are, in my opinion, what the future will be for the pharmaceutical industry and how we can help our patients. As you said, I've been involved in a lot of things. That is very true. But more important than that, okay, I think one view of life that I have always carried. So this is my new book coming out this year. It's not out yet. So I said, what will it be like, say, in 20 years, 30 years, 50 years from today, how we will treat the diseases. And so this book is the first time I've done something. So I combined the philosophy and a lot of science with a lot of hope. So I will give you a little bit of a perspective on what I came up with, okay? And it surprised me just as much as it will surprise you, okay, which way we're heading and how far we will go. So first of all, historically speaking, the drug discovery technology or drug discovery itself, okay, goes into what I call waves, okay. There it is, 140 years of discovery in new drugs. Obviously, we all started, let me just take you back 10,000 years, okay. It was very cold. We never had a time to get out. We lived in caves. So when we came out, okay, there was no grains, nothing at all for us to feed on. But also we tried trial and error, what you call anecdotal medicine. And you know what? 10,000 years later, we're still doing it. Okay. Now we are smarter, and I'll show you how we do it differently. But human body is still so complex, okay. And I think those of you who are involved in any part of research, okay, I get amazed every morning when I think about what is my ribosomes are doing today, okay. Can you imagine, okay, what kind of machinery you have inside you? So the fact is that we go through these waves. So what is the new wave? Okay, I'm going to talk about that today, okay. All these things we talked about, biosimilar, those are the products of yesterday. I think there's nothing more exciting left about them, but I think they have left a legacy which is a lot more interesting. So here's the theme of my book. It's called a non-linear perspective. Now, what does non-linear mean? Okay, I think there's a lot of science and physics and mathematics you will find. So human vision, I can see straight, yes. But to look at, if you have a curve and you're looking across the curve, it's very rare that you can see beyond it. Okay. Let me ask you this, okay. Say about 50 years ago, what did anybody think of PCR, which got Nobel Prize, or CAR T therapy, or CRISPR? We didn't even think of it. Okay. In fact, it was not even imagined. Okay. So things happen that are not in the line of our thinking. Let me go back there. So I'm saying that it is difficult to predict. So in writing this book, I studied a lot of books and papers. So here is the bottom line. Nobody has ever been able to predict future in any field. Never. It always fails. The reason is that our minds can only think linearly. We are linear species. So taking that paradigm, I'm saying that what is going to come in the future will be so dramatic that you and I today talking about it, okay, may not even have an idea how far it will go. So I'm going to start with a very interesting concept, okay. On the left-hand side, you see, say, linear and non-linear. This is a very famous experiment in physics. Lorentz got Nobel Prize also, okay. So what happens if you take a pendulum and you hang it in the air and start, it will keep going back and forth, right? No issue at all. But if the pendulum has another pendulum attached to it, they call double pendulum, and you move it, okay. Now, with today's computing, with today's understanding, you will say that, yeah, I'll be able to predict the movement. But you know what? Nobody has been able to predict what the movement will be like, period. Because there's a randomness involved in there. And Lawrence came up with this equation, okay, which says that this double pendulum, if I trace it every time, the tracing in the 3-dimension comes out like a butterfly, okay? And that's where the Lorentz butterfly effect came. Basically what it says, if there's a butterfly in Brazil fluttering, would it change the weather in Mumbai? Answer is yes, okay? The reason I'm telling you these things is that our understanding of non-linearity stays unexplored. So if anybody goes out, including yourself, myself, predicting what's going to be like, okay, it will be only a wild guess, but no more. So with that in mind, okay, let me show you just very quickly what is going to be in the near future. Okay. There's no doubt about in the minds of anyone who is here, 190 people plus all the administrators here, that biologics will supersede chemicals. Chemicals will still here. Today, 70% or more new entities that FDA is approving are biologics. So what will happen eventually? We'll have more biologics than chemicals approved in the future. Number two, which I want to talk about today is and I think we had a very nice discussion earlier also, nucleic acid vaccines, mRNA vaccines and other things. Okay. Last year was a dramatic year, and just the approval of these vaccines has changed what we will do to use these vaccines. These vaccines were based on nanotechnology to deliver proteins. Would we be able to deliver insulin by PO or by transdermal? I think we may be able to do it. The AI and machine learning, I think there is no doubt in anybody's mind that without those supports, okay, we cannot do high throughput screen. And I'll show you a little bit about it. Okay. And then how about if we are able to synthesize antibodies completely? It's a huge molecule. Is it possible? If I say no, it's not possible, then I'm thinking linearly. I don't know what the technology will be, but I do have some idea because I'm using a new technology for my vaccine, so I'll share that with you. Dr. Woodcock wrote an article that says our clinical testing system is broken. I think in silico modeling will be able to predict much better a clinical response than actually getting into patients. Gene and cell therapy are here. I mean, there are products approved. There's no doubt about it. And so are the early curative testing. How about if you have a testing that's a diagnostic as well as curative? We'll talk about that in cancer therapy. The old science of nutrition microbiome is not going anywhere. In fact, there's a new awakening about how we can reinstall these concepts in our therapy. Personalized therapeutics absolutely is up there. And 3D and additive manufacturing. FDA issued a guideline on 3D manufacturing also. I'm not adding here things like, digital therapeutics and others which are obviously coming to the future. So first I'm going to take the less exciting thing, then I'm going to end up with the most exciting part. So as I said, okay, on the right-hand side are the number of biologics, new entities approved. By the way, as of today, FDA has approved 1,600 drugs only. Okay, and in the last 10 years, okay, more than 50% were biologics. mRNA vaccines, I think, is going to be the talk of the town, not because their ability to prevent infections, but what they can do for other diseases that I will talk about. Antibodies to treat autoimmune system. I'm a type 1 diabetic. I carry my insulin pump. What happened? What went wrong? My own antibodies, I call them rogue antibodies. they start killing my own cells, And that's exactly what happens in so many autoimmune disorders. What if they were to find the rogue antibody and make an antibody to catch that antibody? We should be able to treat, and that's one of the discussions of point I have. I think biosimilars, as of today, there are more than 100 biologics with no patents, with a much lower barrier available to become biosimilars. Unfortunately, all the big pharma and small pharma, everybody's chasing down the big apples. Okay, and my advice to everybody is okay. Go to every biologic drug has a market and a need. So instead of looking at is it a $20 billion drug, I'll take something which is a $50 million market and develop it. And I think there's a need, especially for companies in India, to diversify their portfolio significantly. I have seen the portfolio of most of the major companies, and sort of just everybody is tracking down the same molecule. I think it's about time. Gene and cell therapy is here. There are a lot of products approved. One prediction I'm making today is that the biosimilars will become like generics. My recent papers that I published and submitted to FDA says that clinical testing in patients, now when I say not the PK/PD, but the efficacy testing of biosimilars in patients is totally useless. And I presented my mathematical arguments. And you know what? I'm making a small sense to FDA, okay? I said you should test it if it gives you a reason for any more validity. So I presented an analysis of every biologic approved or biosimilar approved by FDA. So let me just give you a single liner. Okay? They did not allow efficacy testing unless the PK/PD met fine. So they would not let you do that. Move forward. Okay. None of the PK/PD failed. There was a couple examples they redid the study. Then they came to testing in one indication, and none of the study failed. So as if I were Aristotle, okay, I will say like this, okay. If a study never fails, it means two things. A, that the products are similar, which was the purpose that you determined through PK/PD study. Or the testing is not sensitive enough. In both cases, it does not help to add this clinical testing that may actually end up approving products which are not biosimilars. So I was able to get a waiver for 4 major products, no human testing, because that's where the big money comes. Okay, imagine if you have anti-cancer antibody. Okay, how long will it take? Okay, there's no naive patient available. So is it necessary? No. In silico approach is where we have to go and challenge. So as you know, FDA allowed removal of all testing of insulin for immunogenicity. Because my argument was that if immunogenicity does not affect PK/PD, it doesn't matter. 60% of people who get insulin show immune response. So what's the difference? They agreed. Anyway, I think there's a lot of opportunities in biosimilars, but I think the company have to start thinking differently than they do today, okay, and create a more robust regulatory plan to get in there. So very briefly, as you know, today, our current generation that we are talking about, in fact, there was a mention of ADCs earlier today. That's really where the future is going to be. But the next generation, which is about on the horizon, are also very different. How about if you modify the PTMs? Okay, you altered the pattern to give it a better nanobodies or biospecific antibodies, new scaffolds. So now we're getting into an arena of where we can tailor it. And there's no reason why we shouldn't be able to get a better response. And also in applications which were not possible before. So for biologic drugs, a lot is coming in the future. And therefore, I mean, all of them eventually expired, the patents and everything. So I think for many generations, definitely I won't be around, okay, but there will be new biologics, new biosimilars brought out with a lot more effectiveness in the future. Gene therapy, I don't have to tell you much more about it. I think people who are in this room definitely have a fairly good idea how does it work. Okay, we're just trying to change the gene, okay? Fix your defective gene. Now imagine we would never have been able to think about it when we discovered penicillin, did we? No, it was not in our minds that we could do this. We didn't even know there were genes there. So this is what I call the shift that happens, okay, in our thinking. So keep an eye on gene therapy products, because right now these products are so expensive. I don't think any developing country can afford it, period. I mean, if you have to pay $100,000 for a dose, okay, or $2.4 million for one drug per dose, it's just not possible. Okay? And I think it's the responsibility of all the developing countries and companies that I see in India to move on to it and see if they can make these therapies affordable to their people. Very important. By the way, it is not a very expensive exercise to make gene therapy products. In fact, it is much cheaper than making biosimilars. It's just that there is so much IP involved there, okay, that companies are making it charge whatever they want. Novartis charges $2.4 million a dose. Well, you know what? That's only drug approved. What are you going to do? So your good old CRISPR technology, there was a Nobel Prize given in 2000, I believe 19. I don't again have to go into detail. I think everyone here, you can. Today the Google has become so common, okay, that I stopped using teaching things that you can learn from Google. Okay, so CRISPR is here to stay. It has brought so many new tools. So this is one area where again I will encourage the companies in India or other countries to start getting into it. This is the technology of today. Biosimilars are yesterday. Now, as I was mentioning earlier, that in silico drug discoveries and artificial intelligence and machine learning, I don't know how well versed the audience is, okay, but the whole meaning of artificial intelligence, actually I don't like the term artificial intelligence. The intelligence is real, okay? So I suggested use called digital intelligence, but I think people just don't like to change things, okay? There's nothing artificial about it, okay? It just, intelligence is not necessarily a very good thing, I mean, not the best thing. It's just putting the data together and coming up with conclusions. Here's a very good example of, I don't know if you have ever looked at the Bayes theorem. I have a whole chapter on Bayes theorem in my book. It's called Posterior Probability. So on the left hand, you see how to discover drugs. Okay, we apply posterior probability. What happens if you give this dose? What is the p-value? For example, if you, let's say you got tested for COVID and the test came out positive, all right, fine. What is the chance that you are actually positive to COVID? Okay, now this is what the Bayes theorem does. Okay, so it takes into account the false negatives, false positives, and the incidence rate. Let's say if there is a cancer which is only 0.1% or 0.01% population has a cancer. So before you test it, your probability is same as 0.001%. Then if there's a 20% risk in, not risk, but a false negative and false positive, so you can calculate the real p-value and you keep going as you do more steps. And we have been able to discover some great targets, some really good targets, okay, by in vitro studies, just by using a Bayes theorem of posterior probability. But you can see the complex graph. I don't expect you to go through and look at it, because it simply shows, there are thousands and thousands of these tests and links we can combine. And that would require your AI component in there, the machine learning, to tell you have a good target or not. Today's drug discovery, as we all know, has become so well dependent on AI and machine learning that any high throughput screening, you go through the medicinal chemistry, pharmacology, and biomarkers. Those are the key things now. Okay? But it has become so well organized now. We have, I was able to count about 400 databases available, open databases, for you to draw the data and put it through your target identification and everything. Okay. So these are very exciting times. With all the computing power we have, I think we still need more computing power to connect millions and millions of these target molecules. And eventually, I think the goal will be to create a biomarker, okay, that will be indicative of a clinical response, so that we can obviate testing in patients as much as we can. Now, I'm going to talk about something which is the talk of the town, which is called virus. So I was giving a talk, again, this was a Zoom talk to a high school class. Okay, I do a lot of volunteer work. And I said, you and I are viruses. And that's a fact, okay? It was the virus. No, virus is nothing but a chemistry, okay? It doesn't have a mind. When people say the virus mutated, it did nothing. It cannot think. It has no ability to think. It's a process of randomization, okay? So this is what happened. Before the life began, we had viruses. Virus was simply when you put together the nucleotides enough in an order they can replicate. They go through the ribosome. Okay. Now, when the viruses were formed, there were no ribosomes. Okay, and the ribosomes came. In fact, if we did not have viruses, we would not be able to survive as a species. So, if the COVID virus mutates, why are we surprised? Okay, it's not that the COVID virus has a mind to hurt us. Okay, this is how the randomization of chemistry goes. And all the hierarchies and eukarya, bacteria, and we are evolution part is coming from nothing but viruses. So as you know, in October 12, 1768, I'm sure nobody was there in this meeting, okay, the first vaccine was given, okay, and then on December 11 in the morning, FDA authorized BioNTech Pfizer vaccine. And I want to introduce to you a concept. This is Robert Kuhn, who created the vocabulary called paradigm shift. Now, what's paradigm shift? Our thinking, our modality of work, whatever you do, shifts, but only if there's a crisis. So the first thing required for paradigm shift is there must be a crisis, and there must be an available solution, and then there must be a willingness for us to do something about it. I think somebody else mentioned it takes 10, 15. One vaccine took 26 years to get approved. We had the vaccine in less than a year. When Moderna got the gene sequence from NIH, it took them exactly 4 days to have the vaccine ready. Can you imagine this process? Now, RNA vaccines have been around for a long time, but the paradigm shift forced them to come out. And you know what? They came out really good. I don't think any vaccine is as effective as RNA vaccines are. The approval testing by FDA says if you have 50% effectiveness, they will approve the vaccine. So this was a true paradigm shift. Somebody already talked about how RNA vaccine works. It's simply you're putting a chemistry in your body that makes the antigen. and then the show goes on. Okay, instead of, so I've been asked so frequently, what is the safety of these vaccines? I said, these are the safest possible vaccine of all types, because there's nothing in there that can infect you. It makes an antigen and then it dies off. Now, as opposed to this vaccine, if you're doing a DNA vaccine, now there is the issue, because what happens is that the DNA has to work through the nucleus. So I'm predicting today, before this audience, that a DNA vaccine is dead. I wouldn't want you to work on DNA. Even though there were a lot of cancer candidates, RNA is the only way to go. A, it is safer. B, it is a lot cheaper, okay, to do. And third, okay, you can do a very large quantity with a very small investment. So I've set up a new company. It's called RNA Therapeutics. We got some funding from the US government. And we were able to create an HPV vaccine, okay, in less than 6 months. It's very straightforward, okay. There is nothing in there that the group that I'm talking to doesn't know how to do it, okay. It's just simply you have a protein, you take the genome sequence, you make your plasmid, and you just take on the RNA. The production part of the RNA is all in vitro, so you don't have any cell debris in there, okay. But there are some new technologies coming up that I will talk about today, is that plasmid, which is the most common way of expanding the DNA, may be obsolete. Okay. There's a one company I'm working with now. We do 100%, we don't do plasmid at all. We're using what? PCR to make the DNA. And I think the reason it was not available because of the cost was very high, but now the cost has become very acceptable. So we may not even have to use DNA anymore. This is what the RNA looks like. Again, I'm not going to talk about, the only thing I want to talk about is that you can easily get, if you're using plasmid, about 2 grams per liter. And the dose of the Pfizer vaccine was 20 micrograms, and the Moderna is 100 microgram. If you make 1 kilogram, okay, you can make 33 million doses. You know, can you believe it? And you can sell it them easily for $300 million. So all of my dear Indian companies, please think about it. Okay, I think future. Let's get into RNA therapeutics, not just vaccine, therapeutics. So here's a very good comparison. Okay, so this is my company. I just launched last year. Okay, we got some funding from the US government. I'm using PCR and rolling DNA. I'm not using plasmid. You know what that does? That cuts down my capex and opex by 70%. I'm putting multiple RNAs in one vaccine. One day I'm going to be making a vaccine for all STDs together. You got HPV, HIV, all of them together, okay. One of the things we can do now, we would never be able to do before, is that we can create a prevention of autoimmune disorders. There's no treatment for Parkinson's disease. You know why? Because the body itself is acting. Today, I see a bright light. Okay? We will be able to prevent all autoimmune disorders, which present a significant part of world health issue. And this is the area where I would like the new companies or new ideas to go and to look into that. Okay. To get the IND approved on the phase 1, I think 6 to 9 months is all I would say it will take. But there's a huge market waiting for those of you who make sure that there's a money at the end. Okay, so it's not a bad idea, by the way. One of the another areas that became very popular with this vaccine was use of nanomedicine. We were able to put into LNPs to put the particles inside the body. I think there are so many new applications coming for nano. It is probably one of the most budding areas of research. So here's the nano system which is used for cancer they call theranostics, therapy and diagnostic combine. Okay, when you're looking into cancer, you're also treating it. Okay, you can only do it with the nano system. Yeah, I'll be done very quickly. This is my last slide. And here is the thing, okay, that I'm going to show to you is that you have a nanoparticle delivery through intestine. I think we should be able to put insulin through PO route by using nanoparticles. And so that is the last my slide is that past, present, and future exist same time. You're all young people listening to me, okay? Think positive about the future. Look for the future technologies if you can think, and work hard to bring it to mankind. Thank you.
Thank you, Professor Niazi. It was really a nice presentation. I mean, we could still see a sense of amalgamation of science and philosophy in a bit in many places, and that's really it. So we do have got certain exciting questions in the chat box, and if you are okay, I can we can take a few of them, and if time permits, maybe more.
Sure, absolutely.
Okay. So I think one question.
Yeah, go ahead.
Okay. Okay. So you want to take it up? Otherwise, I can start taking some of them.
No, I can see them, so I can go quickly. It's the thing to biosimilars, the drug I yesterday. Yes, you know what? Biosimilars are nearly not. There's nothing. It used to be when in 2014 when I wrote my first book. I was very excited. Okay, but now I see there's no big deal. Okay, the science has progressed. Okay, you should be able to make good biosimilars. There's no excuse anymore. Okay. The Moderna vaccine is lipid technology. Why is it approved so early? Very good. Okay. So Moderna vaccine and Pfizer vaccines both are mRNA vaccines. So let me just give you very quickly how it works. Okay. By the way, I was part of the FDA team also who looked at the data. So what they have to do is to bring in 150 infections in the control and treatment group combined to close the trial. Now, what happened in the US when the infection rates went up, they were able to recruit more people and they came up with 150 infections. Okay. And here's what happens. Okay. So what you do, they had about 45,000 subjects. Half of them got control, half of them got the treatment. Okay. And after they had 150 total positive COVID patients in 45,000, they stopped the study. And now they open the data and find that all of 149 were the ones who did not receive vaccine. Okay, so that's the one reason. Okay, is that there was enough infection. So if I am making a vaccine which has a very low infective rate, okay, it may take me years to prove it. Second thing is there is no toxicity at all. Period. Okay, in RNA vaccines, it's a piece of chemistry going in and destroying itself. Okay, so there was nothing that they had to you have to do to show their safety. It says the question is, when do you think biosimilar become generic? And you know what? They are now. Here's the reason. Let me tell you this, okay? So you can, and remember it, okay? Every applicant who went to FDA for biosimilars offered to do clinical efficacy testing. FDA did not ask. So you know what? There was one big example in Europe, okay? One company was developing EPO, and they said, we are going to test it in 500 patients. What do you think EMA will say? Oh, hallelujah, go ahead, do it, okay? You have to go to FDA and tell them why you do not want to do a study. Remember in the BPCIA it says, and let me quote it to you because I remember so much, if there remains any residual uncertainty, additional clinical study may be required. Nobody asked them what is the uncertainty. You go to FDA and you offer a protocol, FDA say, yeah, fine, do it. So today, I think you should go and challenge them. They know I'm not going to do the efficacy study. This is the reason. But you have to be proactive, okay? Thank you, all of you who put good words, very informative. DNA, Rna Vaccine, let me see. Liver injury increase incidence of HCC. Okay, well, liver injury, as you know, okay, there's all the enzymes and everything are there, okay. Then Divya is asking, what is the cocktail of antibodies? It's very simple, like the cocktail says, okay. You can have a straight whiskey or you have a cocktail, okay. You put many of them together, okay. Exosome-based delivery, I think it's a fantastic idea. This is coming up, but keep in mind, you know, to get through faster, okay, I would like you to stay with more conventional. For example, somebody was mentioning today that the yield of the cell line, 2 gram or 5 gram or 10 gram, I say it makes no difference. Let me tell you why, okay? Your cost will be depending on the carbon in, carbon out. Mammalian cells are living entities. If you push them hard, they go crazy. You have a much better product at a lower yield than at a higher yield. Why would it matter, okay, if I'm getting 2 gram or 5 grams? Okay? It does not add to the cost. Okay? Most people are just getting so swayed by high yield that they are compromising the quality because of that, okay? Let me see. Academic institutions, you know what? The top 4 biological drugs, okay, today were discovered in academia. I think academia can do, you remember I showed you a lot of possibilities. I think that the industry can benefit greatly from academia if they get into, you know, fund them and let them come up with some creative ideas, okay? So here we go. Let me see. Okay, let's see. Kindly share. Okay. I think, is there anything else that I need to talk about?
So one question from my side, Professor Niazi.
Yeah.
So
Sure.
we have seen a lot of biosimilar opportunity. Definitely the Indian companies are taking up and hopefully they will do more. But when we talk for this cell and gene therapy from the Indian perspective, especially when this space is highly kind of protected by IP, maybe we may not be the innovators. We are still behind in that game. But what do you see as a future for our, I would say, in a contract manufacturing or a partner for a lot of supply for a lot of the cell and gene therapy like viral vectors, etc. I mean, where do you see the future for developing countries?
Very good.
I mean, like
I
India.
think, yeah, no, I think you're absolutely right. Okay, the same issue was with the with biologics. Okay, there will be an IP involved, but eventually everything comes down. Okay, now here's the part of it. As an IP law practitioner, okay, if you show me a patent, I can show you how to go around it. Okay, because here's the reason. One of my argument with the big pharma was we sued the FDA saying that only we know how to make it. Okay, so I told them what you did was a method, not the method. Okay, so I think there is so much room for creativity. You may not end up getting your own IP, but you can definitely, you don't have to infringe any IP to do all cell therapy and gene therapy anymore. The reason is that same thing happened with the biosimilars. If you look at the early biologics, there was not much IP involved. They never thought of it. Now, you know, adalimumab has about 400 patents. So I think my idea is if you want to get into gene and cell therapy, you should get in there and find out how do you go around it, okay, which is not difficult because there's so many ways, different ways of doing it, new technology coming up, okay. And same thing applies to biosimilars, okay. I think there's a need to take it on a more on a generic side, okay, rather than going through the expensive trial basis.
Thank you, Professor Niazi. That was really, really great insight.
I know my time is up, okay, otherwise. But you know, you have my email address there. Just or on the LinkedIn. Feel free to connect with me, okay? I love to talk about it and I love to give you whatever I have because I really think there's a future is very bright for us, okay, in our field.
So we received one more questions, and I think what I'm
Sure.
getting from the organizer that we do have some time, so we really are not pressed for the time.
Okay. I have no issue. I can stay forever.
So I mean, this is very basic questions, but maybe it's good to get your perspective. What's the difference between generic and the biosimilar drugs? And probably if you can elaborate, there are a lot many people from academia, and maybe they will be really interested to know that.
I think it's a very good question. Let me tell you why. When I wrote my first book, I called them biogeneric therapeutic proteins. Okay. Then when the FDA started working on it, I got involved with the FDA. Then they explained to me why the word generic cannot be used. It has a legal issue. Generic products were automatically substitutable at pharmacy level. This was the US issue, okay, so not the global issue. So they say we cannot use the word generic. So what do we use then? So I tell you what, we had a meeting, we talked about many different words, okay. So let me tell you what WHO calls, okay. They call it similar biologics, same thing in India. I totally disagreed with it. I said, these are not similar products that happen to be biologic. These products are biologically similar. They could be different chemically, biologically similar, not similar biologically. Okay. So we agreed on you word biosimilar. Exactly same thing I did when most of you were not even born. Okay, when the generic law came in early 80s, we call it bioequivalence because these are biologically equivalent, and that word is stuck. Okay, so when I say generic, I simply mean that in a generic product, you do not have to do efficacy study. So when I say biosimilar becoming generic, it doesn't mean, at least in US context, that they will become substitutable, because there's another category called interchangeable biologics, which nobody has filed, nobody will get approval. I think it makes no sense, okay? So the point is, I think today you have an opportunity to challenge all efficacy studies. Let me share something with you which may or may not be liked today. I've reviewed many studies that were conducted in India on biosimilars, clinical studies. The only thing I can conclude was the study has no meaning. Okay, every study must have a statistical protocol, have a power of the testing. Okay, just because you put in 100 subjects, 100 patients, and nobody dies, okay, it's not a clinical trial. You're better off not doing it. Okay? So what I'm saying is that I think it's up to the companies who are developing to take that scientific approach. If you can do it, and you know, in the new FDA plan that they call, there is a word used called in situ. You can do in situ testing or in silico. Okay, I would rather do a PK/PD study that takes care of everything. It takes care of multiple dosing, takes care of immunogenicity, everything all in one, a comprehensive single study, and that's all what will be required. I don't think we are going to remove that ever. Animal toxicology, totally useless. It doesn't do anything. But the new FDA, if you're engaged in it, I'm not too familiar with CDSCO. I think their guidelines really don't make any sense anyway. But my point is that if you want to call them as generics, okay, you have to develop a portfolio that does not require testing for efficacy.
That's really helpful. I think we do have one more questions on how do you see a future for cell and gene therapy in next 10 years?
Oh, here's the part. So when I showed you the first slide, okay, what amazes me is how incompetent I am in telling the future. And so is everybody else in this universe, okay?
Right.
You know what? The moment you say, I know what's going to be happening tomorrow, you are denying God, okay? I think we shouldn't do this, okay? God is a pretty funny man, okay? He can do funny things, okay? So today's cell and gene therapy is here today. Okay, I think we would not be able to prevent all diseases or all therapies. We are now getting into an arena where we are actually modifying our DNA. You know who else does it to us every day? The viruses. You have millions and millions of different viruses in your body today, as we are talking about it. So I'm not wearing a mask, okay, so you cannot get from me, okay. But by the way, I got my vaccine. I got 2 doses of Pfizer. So I'm done, okay, no side effects, okay. So yes, cell and gene therapy are today's technology, not tomorrow technology. Tomorrow we will be talking about most likely an automated system, okay. Diagnostic is gonna play such an important role. I could have my gene tested. By the way, we were able to finish the genomics about 10 years sooner than we were planning, you know that, okay. So we have complete human gene, okay. We will be able to find out where is the issue. And then we will be able to administer RNA therapies or CRISPR therapies to fix these things. Okay. Today, the challenge is I don't want you to go and start changing the DNA and create another human being, okay, which is totally different than you and I, okay. So the question is, can we create another species of Homo sapiens? The answer is absolutely yes. Okay, should we do this? Okay, I don't know. I think let the nature take its course. Okay, I think the nature takes its time. Okay, there's a book. Okay, it's called the Homo Deus and Homo. If you read that book, okay, Homo Deus, okay, you will figure out, okay, what is happening, will be happening. I think we will have a lifespan of 150 years if I can stop the fracking of DNA. Okay. My question is, okay, what would you do with somebody living for 150 years? Okay, would they be working for 100 years? Okay, I think it's better that we leave this world, okay, when the time is for younger people to take over. Okay, so there are a lot of questions about this, but gene and cell therapy is not future, it's today. It will be much expanded tomorrow in modifying most of our genetic diseases. BRCA2 gene will be the first one to go. I predicted in this book, you know, in 20 years, so we will have no cancer. There's no need for it, okay? It's basically an autoimmune disorder, okay? Our body is incapable of taking care of the mutations that are happening in our cells, okay? If I could fix that, you know, cancer will go away. There's no doubt in my mind.
Great, thanks. There is one more question, Professor
Sure.
Niazi, and this comes from the academia side. And since you have seen academia, you have seen regulatory, you have seen industry in US, how do you see the academic institute in India can play a role? I mean, where do you see the gaps? Where do you see they can play a role towards the innovations, towards having more partnerships with academia, especially in the field of biosimilars, and making them more cheaper and affordable to society?
Very good. Okay. So I have a small model to share with you. Okay. So my new company, what we do is that we develop a vaccine, RNA vaccine, and only take it to phase 1 IND. It takes about 6 to 9 months. Okay. Then we license out. I don't want to get involved with it. Okay. And I don't have the resources to take it through clinical. Okay. I think this model is so well suited for academia. Okay. Let them. Let's say there's a new biosimilar to be developed, okay? They can bring it to the stage of analytical similarity testing. And once they have that testing, okay, they can hand over this to the industry. And they can do it much faster. Let me tell you something, okay? The big pharma today survives at the shoulders of the small pharma. Adalimumab was not developed by Abbott, okay? So I think academia can play a very significant role, but only keep in mind, okay, the industry has to justify the expense and cost, okay, which is very fair, okay. If you're not creative, you cannot show the value. Don't dig into science too much, okay. One of my talks in India was, do not invent anything new of a higher end. Do not invent a new chemical or new things. There's enough technology available. Okay. Japan is the worst country for science. They have the lowest number of Nobel laureates per capita. They don't give a damn about science, okay? They would develop technology, okay? And I think every developing country must look at the technology deployment, how you can serve the needs of the country with technology. So academia, if it takes a creative path, and I'll give you a good example, okay? I'm using a PCR to produce DNA, not plasmid. This is the first company that ever did it, okay? Nobody has done it before. I went to FDA, they say, I have no issue with this, okay? As long as it looks like DNA, I'm okay with it. So what I'm saying, you gotta take a step forward, okay? And not just accept things as they are, status quo. This is how it is done. When I had my company, I hired over 500 people. I used to always fire somebody who will say, this is industry standard. I said, who makes this damn standard, okay? Okay, I want you to tell me why are you doing it, okay? So I think one of the myopia comes in industry, in academia, is by sticking to the books too much, by not being creative for the end part of it. In the end, you have to produce a product that can be sold, a technology that can be used. And I think industry can benefit from it. It has to be a two-way, and I think you get a much better ROI if you invest in academia rather than even your own laboratories.
That's really interesting proposition. I think
let's do it, man. I think we need to. India is so far. I'm so pleased with the progress in India. I think it's about time to take this paradigm shift, okay? All right, and get into the real science and leave the inventions behind. Just take the technology and make the best out of it.
Thank you, Dr. Niazi. I think that was really helpful. Okay. Very good insight into many areas, and I think people will keep hearing you probably for the whole night. But we do need to end this session at some point of time and definitely look forward for the next opportunity. And I hand over to the organizer. Thank you, Professor Niazi, again.
Thank you. You're welcome.
Thank you, Professor Niazi. I think he has left immediately.
Oh, okay.
Yeah.
Maybe someone want to reach him out, or
yeah, if he can join us back.
Maybe
I hope
some of the organization
we have to honor him with a vote of thanks and the certificate. I think
it's up to the organization team if they do have contact of him and whether he can.
Yes, we do have contact. We'll try to. But meanwhile, let me complete few of the formal instructions for the participants. So thank you, participants, for attending the day 1 sessions. The feedback link as well as the quiz link has been sent to your email, and you are supposed to reply to it by tomorrow 12 noon afternoon, Indian Standard Time. And we could not have, because of some unavoidable reasons and important meeting of Dr. Parth Shah, his lecture today. So we would reschedule his lecture in the coming 3 days, and we will notify you at the earliest. At the outset, we are very much thankful to Mr. Dhaval Trivedi for nicely managing and handling the scientific session, which was delivered by Professor Sarfraz Niazi. And we at LM College of Pharmacy are very much thankful to Mr. Dhaval Trivedi for spending his valuable time, giving his consent to chair the session at our E-International Conference. We know the college is our college is established in 1947, and we have someone who is pioneer in biosimilars and bioequivalence, a huge personality like Professor Niazi. So we are honored. It was an honor. It was a pleasure for all of us at LM College of Pharmacy to host Professor Niazi, sir. We have seen him through his extensive work, through his published articles. We have celebrated his accomplishment when we were studying our undergraduate or postgraduate in his books. He is the man who has been revolutionizing the health in this world. He is someone, a scientist, who has given a pathway for approving the biosimilars. And if I would not exaggerate, the FDA has listened to him as well. So it was a great learning treat and learning feast on day 1. So let us call it a day. And I request my colleague, Dr. Radhika, to honor our speaker, Professor Niazi's certificate and Mr. Dhaval's certificate. Thank you very much, Dhaval sir, for giving your valuable time to all of us and handling the session in an effective way.
Thank you. Thanks. It was really my pleasure.
Thank you. I request Yamini madam to please virtually present a certificate to Chairperson Mr. Dhaval Trivedi.
Mr. Dhaval, please accept from our side your certificate for chairing this session. I thank you again.
Thank you. Thank you.
And dear participants, we have with us Dr. Hemant Damne. We welcome him over here, who was to chair a session, today's session of Dr. Parth Shah, who is from the fellow hematology from Harvard. But because of some unavoidable reasons and his emergency meeting, he has to leave. We are going to reschedule his lecture in the coming 3 days. So we'll have you back again, Dr. Hemant Damne, who is a senior scientific officer at Immunol Therapeutics, Bangalore. Thank you very much, Dr. Hemant, for sparing your valuable time. And I hope you also have enjoyed the 2 days, both the scientific sessions. So that's all from our side at Allen College of Pharmacy. And we request you to fill up the feedback form as well as the quiz, which has been emailed to you and reply to us by the 12th afternoon tomorrow. Thank you. Do stay safe and take very much care of yourself. Good night. Thank you.
Thank you.
Thank you.
Thank you all of you.
We will be back again tomorrow, live again at 4 p.m. You will have to log in through the same participant link which you have logged in today. Thank you very much.